Okay, I'm going to do a little video here to go over chapter 1. Let me get this going here. All right. So chapter 1, again, is our introductory chapter. We're going over sort of where we're at in terms of life and what humans are, and an introduction to what science actually is. All right, so that sort of sets the stage for the rest of the course. So we began by questioning what is the difference between living things and non-living things. All right, now the thing to remember is that both living and non-living things are made up of the stuff on the periodic table, right? But in living things, those things on the periodic table can be organized into special molecules that you will not find in non-living things. Now, in living things, of course, we still find our elements, right? We have our atoms, but there's also these 4 biological molecules that are unique only to living things. And we are going to go over each of these in great detail, or in better detail, not great detail, but much better detail as we move through the course. So for now, I just want you to remember what these are and sort of, touch base with yourself and try and remember from way back when you were learning this in high school what these things were. All right, so first of all, we have proteins. And like I said, we will go over it again, but I just sort of mentioned in class, tried to jog your memory, what are proteins made up of? And some members of the class could remember amino acids, okay? And we'll go over what those are. So we have proteins. And then we have lipids, all right? So lipids is a sciencey term for fats, okay? But when it comes to fats, there's actually several different types, and we will go over them. And I mentioned in class one of them that you've probably heard of before, and that would be the steroids, okay? Then we have carbohydrates. Carbohydrates are sugars. Now we have a variety of different types of sugars, some little small ones and much bigger ones. But again, the carbohydrates is the 3rd category, and those are basically sugars. And then finally, nucleic acids. So nucleic acids, those are the large molecules that include both DNA and RNA. So we are molecules of heredity and to facilitate protein making machinery. And again, we'll go over those just very briefly in this course. We spend much more time on the first 3 than we do on this 4th one. But anyways, we will remind you the differences between DNA and RNA a little bit later, maybe in a couple of weeks in the course. All right. So those are our 4 biological molecules, and these are unique to living things. Now, the second thing that is common to all living things and not required by non-living things is that we require energy and we require raw materials. All right, so we get our energy ultimately from the sun, but we get that basically from our food, all right, because we are consumers. You can remember that term. So energy, and what is our energy currency? We'll remind you this many times in the course, but if you think back to when you studied this before, it's ATP, all right? So we will talk about ATP a little bit later. What are our raw materials? Well, it's the food and water that we take in. All right, so these are required by living things. So we combine energy and our building blocks or our raw materials, and we get life. Now, the next thing is that living things are made up of cells. Now, you can have complex living things like this puppy and the flowers and the grass and the tree, and you can see these are all things that we can see, macroscopically. These are multicellular living things, right? We're made up of more than one cell. But there's also a whole slew and population of living creatures that are unicellular, all right? They are just one cell, right? And we have trillions of these in our body, and we need them for our life. The bacteria, okay? But again, even though they're just one cell, they still meet all the requirements for life. Now, in class, this is not something I'm going to put on the exam, but in class, I mentioned that there are some forms of creatures that science isn't quite sure about classifying them. And the one I mentioned were prions. Those are, if you want to look this up, if you're interested, I believe it's spelled Prions, prions. And these things are able to reproduce, all right? They're responsible for certain diseases. It's believed that they might have a role in Alzheimer's, but definitely they're involved in things like mad cow disease and scabies and that kinds of things. And these things are not made up of cells, and they are, able to reproduce themselves. The other thing that I didn't mention in class is just much more familiar to you are viruses. Viruses are not cells, yet they are clearly able to do everything they need to multiply. So this is sort of where science is sort of on the bench about classifying these. And obviously these 8 categories are sort of been out there, and there are these little, other things going on in the world that scientists are debating and how we classify them. All right, you might be surprised by that, but I'm just throwing that out there. I'm not going to put it on an exam, but I think that you should know about these things. All right, now, number 4. This is a big thing in this course. Living things, all right, maintain this thing called homeostasis. Homeostasis, a really big, important thing in this course. So what is homeostasis? And really what this is balance, okay? This means that living things need to maintain some sort of stable environment within themselves. And that is what is conducive to the life of that living thing. Now, this does not mean equilibrium. All right, so homeostasis means balance, but balance does not mean equilibrium, especially when it comes to living things. Living things actually are spending lots of energy fighting equilibrium, because once an organism is at equilibrium, they'll die. Okay, so I'm going to show you what I mean by this. All right, so in order for living things to survive, they have to maintain gradients of concentration, gradients of fluid, gradients of all kinds. So gradients is a difference, all right, on either side. And the reason that this is so remarkable in living things is that living things are open systems, and that's what I'm sort of showing you here, okay? We have an outer covering, right? Even a single-celled organism has an outer covering, but it needs to take in foods and water and salts, everything it needs from the environment, so it's sort of open, it takes it in, and it also gets rid of its waste, okay? So in that way, it's an open system that needs to maintain a balanced internal environment, right? So we ourselves take in food, water, and that sort of thing. We take in oxygen, and then we get rid of our waste, CO2, and we pee and we poop, right? So these are things that we're getting rid of the waste that we don't need anymore, or that if we've taken in too much that we can't process, okay? Now, when it comes to these gradients, and this is something that you're going to see over and over in this course. So you can see here, we have the differences between the intracellular fluid, so that is what's on the inside of the cell, so that's here in the beige, and then over in the purple, we have the extracellular fluid, all right? So if you look at 3 of our elements here, we have sodium, chloride, and potassium, all right? Now, when you look at this, look at the concentrations. You see that the sodium concentration is much higher on the outside than it is on the inside, as is the chloride, but look at the potassium, it's reversed. All right, so your body spends a lot of energy that you take in from your food to maintain this gradient because it's necessary for life. If these gradients aren't there, these are the ones that fuel your nerves and your muscles, you'll die. Okay, so it's quite as simple as that. So this is what I mean. If this was equal, you wouldn't work, all right? So it's balance, all right? So there has to be this balance that maintains the proper mix of concentrations and everything that we need to survive, okay? Now, I know that's sort of a far out concept, but we will explore this a lot in this course. So that's why I'm talking about it a bit more right now. Now, the next thing is that because we are open systems, or living things are open systems, they're able to respond to their external environment. And I use this little guy here because he's able to do something we can't, but I just wanted you to remember that, right? So this guy, the chameleon, is responding to its external environment by changing its color, right? It's doing camouflage. We can't do that, but then I asked you guys, well, what is something that you can think of that we do to respond to our external environment? And usually the top one we say is maintaining our temperature, right? So that's this idea here, and I'm introducing this slide. We'll talk about it later in the semester, the same slide. I have this reputation of using the same slides over again. You don't realize this, but you will see this again. So, but for now, just remember that we are responding to our external environment, the change in temperature. And we all know what we do if it gets hot, right? That's at the top. We start to sweat to try and cool off. And we'll talk about how that works. Conversely, if it gets cold, what are we going to do? Well, we shiver. We try to raise our body temperature. Or we'll just be sensible and put on a coat, right? Hopefully. But we'll explore how does this work, and how does our control center, our brain, understand what's happening, right? Okay, so then the next thing, number 6, is that living things can grow and reproduce, all right? In that growth, we can change a lot, right? We can't quite undergo metamorphosis like a frog can in terms of its growth, but you do change. You don't look the same as you did when you were a baby, right? You've changed quite a bit. And there's all kinds of factors such as hormones and things like that that are responsible for that, as is what happens with a frog when it goes through metamorphosis, all right? So similar and different. So we are changing, we're growing, and of course we reproduce, right? So we have the ability to do that. And that's all living things can do that. All right. Then number 7, evolve as a species. Now, this is not something that you're going to notice in you yourself, right? That's why it's important to understand. Evolve as a species and adapt. And living things do this very well. And it might take a little bit longer than one lifetime, but it does happen over time, right? And I used in class the example about how the human species as a whole is getting taller, all right? And there's all kinds of reasons for that. And how that's going to play out over centuries, I don't know, but it is something that is happening. And if you sometimes you can read magazine articles about how we might change in the next 200 years just to adapt to our environment. But we are changing. So how we look as the human species now might not be quite how we look in 5,000 years from now if we're still walking this planet. So, we are changing, and that's something that we have to accept. Now, with that, things that we as humans have observed in this process, we've seen things like finches, right? We've seen ancestor finches in the record, and we can see how those have changed. This is largely Darwin's work. He could see that there was different kinds of finches that had evolved to feed on different food. They were all finches, but they had different beak types, and they all could live in the same community and not compete with each other because their beaks made it such that they were specialized eating different types of food, right? So seed eating, woodpecker type of insect eating, and just insect eating, right? Another really wild one that I like are the mud skippers, and these are those fish that can come out on land and walk around, or walk not like we do, but skitter around on their fins. And their adaptations, how they've evolved is that they have a pouch of water over their gills so that they can still breathe through their gills. And if their skin is wet, they can also take in oxygen through their skin. So they're sort of, really cool like that, almost amphibians in that regard. So these guys are sort of in this interesting little niche of their evolution as a species. All right. The next thing is that, and again, that's something we'll talk about a lot, is that living things can metabolize. So that's our 8th one. What does metabolize mean? Well, we'll talk about this a lot, but metabolism is basically made up of 2 parts, all right? We have anabolism, which is where we're making molecules and we're storing energy while we do that. And catabolism is where we're breaking down molecules and releasing energy as we do that, okay? So think about it. We catabolize our food, and then we anabolize the constituents of that food to build proteins that we need, or sugars, okay? So metabolism. So then the next thing is a little just sort of thing, where do we fit in as a species in the entire way that life is categorized on Earth? Now, we are working with a domain system. There's 3 domains. Okay, so bacteria, archaea, and eukarya. And we are here, of course, in eukarya, in Animalia as a kingdom. So you can see here that we've got the 6 kingdoms. Now, I put this little part at the bottom of the slide up here because if you Google, and I know some of you do, you might see something saying 5 kingdoms, and that's sort of out of date. It used to be that the bacteria and the archaea were combined into one kingdom called Monera, but the modern way of looking at this, and the correct one for now, is that we have a 6 kingdom system. So why were these, both these bacteria split into different kingdoms now? And that's because the archaea bacteria, these ones are really primitive. We say primitive. They live in these extreme environments, and they have unique physiologies to allow them to do that. So they live in really, really salty environments, really super cold environments, or super hot environments, or environments that usually aren't conducive to life. So they were separated out from the eubacteria. So these are the bacteria that we're normally experiencing, such as E. coli and that kind of thing. All right. And then on the other end, we've got the fungi and the plantae and the protista. We don't talk too much about those in human biology. We really focus on humans. I guess that would make sense. But here you are. This is where we fit in. Okay. Now, defining what does it mean to be human, all right? So things that make us human. Now, some people like to say, I said this in class, some people think this is what makes us special. I don't consider that as a person. I say this is how we're different. So this is, the defining features of humans, and they are related. Now, I want you to really think about these, all right? Usually on the exam, I ask you a question about this that tests your understanding of it, not that you've memorized these 4 things, okay? So let's think about it. So bipedalism. So that means we're able to stand upright or walk on 2 legs. And with that, we also have opposable thumbs. So it means our thumbs are opposite, opposable, and this is letting us grasp objects between the thumb and the tips of the fingers. We can do this really well. Yes, chimpanzees also have opposable thumbs, but ours are much more fine-tuned to very, very specialized motions. We're a little bit further along in what we can do with that opposable thumb thing. Now, if you think about that, just look at those 2 together. So bipedalism and opposable thumbs. So opposable thumbs lets us carry things and write and, grasp things. And walking on 2 legs frees up our hands in order to do that while we're walking or running, right? So you think about that, how that might have been important to our progressing as a species, right? So we can walk and carry tools, for example. Now, number 3, large brain relative to body size. Okay, it's not enough just to think large brain. There's also animals that have bigger brains than we do, right? Whales and elephants clearly have bigger brains, right? But it's really relative to our body size. It's really just one part of that brain. That's our cerebrum. That's our region of our brain where we have abstract thought. All these things that are human inventions. When you think about it, mathematics is really comes out of that. Even things, our concept of time, right? How we live our life according to the clock, right? That's really something that's in our heads, all right? If you really think about it. But this is sort of why we tick in a different way than other animals. And that's not to say that other animals aren't very smart. They are, but we have this different way of thinking about it. And on top of that, all right, And is number 4, our capacity for language. We have a very complex language as a human species, and it's not just spoken, it's also written. All right. And I had, and think about what that has allowed for, right? So this written comes from that brain thing that I was talking about, right? So there's a lot, other animals have very complex languages. I was saying how I was over at the School of Ornithology in Ithaca, and there's a room there, and I really recommend it if you go there. You can go in there and you can listen to the language of whales. And I mean, that sounds weird because it's at the School of Ornithology, but they do other things there, the biodiversity. And you can hear how complicated their languages are and how different whales speak, even of the same type of whale. So orcas, there's different types of orca, and they don't really mix, all right? And they have their own little dialects, so it's really fascinating. Okay, so it's not that we're the only ones that have a complex language, but we also write it down, right? So how can we write it down? Well, we have opposable thumbs, right? Maybe whales would love to write their language down, but just can't do it, right? So, this is why I don't say that we're special. It just makes us different. And this combination of these 4 things have really defined the human experience, okay? Now, all right. Now, hopefully that you can, you're on my wavelength here about why this is important. Okay, next. All right, this is sort of setting the stage for this course. How we're doing this course, we're going to start off in the next chapter looking at atoms and molecules, and then we're going to move to cells and tissues. So we're going to get bigger and bigger in our experience, and then right into organs and organ systems. And basically our course is going to cover organ and organ systems all the way till Christmas. We'll talk a little bit about the organism as a whole, but the rest of it, population, community, ecosystem, and the biosphere, that you're going to do in other biology classes if you take other ones, right? So we're really focused, we're human-centric in this class. Okay, now the next thing that is really important that we need to cover is science. What is that, right? Well, science really is 2 things. One is knowledge. It's knowledge about the natural world. But the second thing, and that seems to be the thing that causes a lot of problems these days, and I'm not quite sure why, but it's the process. And you've probably learned versions of this process as you went through school, all right? And I'll just say that very often when they teach this, when you're younger, you're not quite ready to fully understand the complexity of this and get kind of muddled in the translation, all right? So we're going to go over these 5 steps, but I want you to understand that there's so many different types of science that use those 5 steps in different ways. So me, I'm a biochemist and a cell biologist, and the way I do my science, even though I follow the 5 steps, it's going to be a little different than how a physicist is going to do the 5 steps, or someone who's a physiologist does the 5 steps, all right? So just sort of take that and develop this overall arching understanding of this, okay? So first of all scientists observe things. We observe and we generalize. And we do this as a human species, right? You're always doing this. You're always looking at things throughout your life. You're observing them, you see them over and over again, and you generalize about why this might be, all right? Scientists just carry it a little further, which is what makes scientists a little different. It's not enough for us just to generalize about things. We want to understand, and we want to know if our gut feeling is correct, all right? So in order to observe and generalize, humans use this idea of inductive reasoning. We're looking for patterns. This is how we survive as a species. We look for patterns and we try to understand them, right? Well, some of us try to understand it more than others, but we do try to look for these patterns, right? So I use this as an example. Every winter, if you're in New York in the past, it's been colder than the preceding summer. So we pretty much know that in the winter, it's going to be colder than in the summer, right? That's not telling us about why or how, but we've observed and we've generalized about this, okay? Now, very often then we'll sort of formulate an idea about why this might be, right? There's all kinds of hypotheses in the history of human civilization as to why it might get colder in the winter, why it's dark at night, right? So it's a tentative explanation about the world, all right? It's based on those observations and that inductive reasoning, all right? And it proposes an answer to that question, all right? And the next part is what's really important when it comes to science. Whatever it is, it has to be testable, okay? It has to be testable, and it also has to be falsifiable. So you have to be able to prove your hypothesis wrong, all right? If you can't prove it wrong, it's not scientific, all right? So these are two really critical things, right? People very often look at scientists and think, oh, well, you're so close-minded, you're not a free thinker. But no, that's not how this works, all right? Scientists, we have to be able to test our things. We have to prove them false if they are, okay? And if we weren't able to do that, we wouldn't have been able to progress as a human species, all right? It doesn't mean that all the other things that are going on in life, that they're not as interesting to scientists. Of course they are. But when it comes to science, these are the things that are qualifiable as a proper hypothesis. It has to be testable and falsifiable, okay? Now, the next step is that you're going to make a testable prediction, all right? Now, this is going to have a little bit more observation, right? It usually involves some sort of an if-then statement. If this is what I think is happening, then that's what's going to be the result, all right? And we're going to use an example of this. So this is usually very well defined. It's measurable and controllable, all right? And this is based upon the next type of reasoning, deductive reasoning. And this is happening for repeated observations. You're building up a set of data or a set of facts, repeated observations that help you come up with different ideas or reasons. Something that's going to help you reach a logically certain conclusion. And it's going to be used to do the next step, and that's designing your experiment, okay? So let's think about this. And you'll see my experiment is using water in plants, okay? I think, most of us throughout our life at this point, even you guys are much younger than I am, but you do know that plants need water, right? Plants need water, and if you give too much water, that's probably not a good thing, and not enough water is also not a good thing, right? There seems to be a sweet spot, and you already know this just from your life, and this is your deductive reasoning now. You've gone through a process, or you've observed other people go through a process struggling with their houseplants, or you've heard on the news that sometimes crops have too much water, sometimes there's not enough water. So this is that deductive reasoning. You've got a collection of facts now that you have to now design an experiment, okay? So let's look at this. This is the experiment. In lab, we do the same thing, all right? I use the same thing in lab and lecture here. So we have our plant, okay? And you look at this diagram here, you can see that there's different amounts of water. So it looks like the idea here is that if I give an increasing amount of water, my plant will grow, all right? So let's think about that, right? So we might say, oh, well, if I give 20 milliliters of water, my plant will grow, but if I give 40 milliliters of water, it's going to be too much. We don't really know what the scientist was thinking about here, but we know that that's sort of what we're dealing with. So there's several predictions that we could make here, okay? So let's say it's this one, just for argument's sake. If I give my plant 20 milliliters of water, it will grow optimally, all right? Too much might be 40, no water, which is my other one, is not going to be good, right? So we have an if-then statement that we use. And then we develop what's called a controlled experiment. And the first thing, the experiment has to be simple, very carefully planned. Don't make it too complicated, right? Don't try to test a bunch of stuff. We're just testing water here, all right? And it has to be controlled. Now let's think about this, right? Think about all the other things that could also influence the plant growth. And the one right here is shown. Light, right? We know that plants need light, especially green plants. We'll talk about that in another course, but right, so we know this. So this is something that we know we have to control if we're really going to test this water. So when we're talking about a controlled experiment, that means everything else within reason, we have to try and keep it the same and only vary the thing we're testing, all right? And that is water in this case, okay? And in lab, we went over the different things that might also influence this, and clearly light is one thing. So we have to make sure that there's always the same amount of light. Now, that brings us back to these variables. So the independent variable is the investigated variable. So I stands for investigated. So what is our independent variable? Well, the amount of water here is what we're testing, okay? Now the other one is the dependent variable. The dependent variable is how you're going to detect the growth of this plant, okay? What are you going to measure in order to see if your prediction is correct? What are you going to measure? There's all kinds of things that you could measure in terms of plant health, okay? The one that I'm using here, I suggest here is the one that most people think of. It's plant height. My plant will get taller if it's growing, okay? But other things you could use, if you look at this as a flowering plant, you can look at the number of flowers, the number of leaves, how big the flowers are, right? How thick the stem is. There's all kinds of other things that you could test as a readout for that independent variable, but you have to pick one, all right? So pick one, otherwise your experiment's gonna get a little bit crazy, okay? All right, now, if you want to look at some more stuff, I have a link here. I'm not going to show, I didn't show this in class, but there's another one also involving plants and the color of light, all right? So, if you understand these and understand your independent and dependent variables, you're good. Now, the last step, and this is the big one, and a lot of people who aren't scientists have trouble with. And this is the fifth one: support or falsify the hypothesis, okay? So the hypothesis can't be proven true. It can only be supported or disproved, okay? What do I mean by that? Wait, what if I have my plant, I'm giving it more water and it keeps growing, so we know, of course, that you give it water, it's good. But remember what I said, what if we give the plant too much water? We know that that's not good, right? And what if my experiment didn't test that? What if my graph line just kept going up, right? It was, a happy plant, but I never experienced that higher end. So the hypothesis is just for that one little experiment, right? And, if you've disproven it, sometimes if your gut feeling says, and we did this in lab with the coin tossing, if your gut feeling tells you that this is the result that I should be getting, but I'm not getting it, then you have to look at your experiment and figure out why, right? Okay, so this is just one little experiment, right? This is why I can't say that this is true. And it's really difficult for a scientist to say anything is true, but we'll talk about that, right? The closest that a scientist will ever get to saying something is true is if hundreds of experiments from many different people come together and they all are pointing in one direction. Then we have something called a theory, but we'll talk about that in a sec. But a theory is as close as a scientist will ever say to say something is true. But the opposite, they'll tell you something is not true because there's no evidence that supports it, all right? So we're going to talk about that now. This is really, really an important thing to think about, especially the way things are going these days, all right? Now, how do we get past this experiment? How do we, get a more solid hypothesis? How do we get to increasing confidence? And this is just done by constantly repeating the experiment in different ways, testing plant height, testing flower growth, testing thickness, trying different amounts of water, different amounts of light, and you develop a whole body of truth, all right, in terms of what do we need for plant growth. And this is done over and over and over. If you're a scientist, you're never done. You never are. There's never enough experiments that you could finish in a lifetime that are really going to fully, allow you as one individual to do this. So this is why you need help. You need lots of help in order to get this done, all right? So how does that happen? Okay, well, scientists are obligated to make their findings known. They have to publish their results. And this is not an easy process, I'm going to tell you, all right? I just published a paper this past spring. It was difficult, all right? Now, why is it difficult? Okay, so you finish a series of experiments, you write a paper, you send it to a journal, all right? Now, these journals, these are not like newspaper journals. These are real scientific or scholarly journals, and in that, there is a panel of experts that review the articles, all right? And these experts carefully go over your results, and they look at how you did your experiment. They look at what they would have done, how they would suggest to change it. And just because you submitted the paper doesn't mean that they're going to accept it. Very often your paper is rejected. And the reason it's rejected is because they feel that you didn't think of something, or you didn't look at something in a different way, all right? So very often you get the paper back, you have to change it, do some more experiments, you send it back if they allow you to. Sometimes you have to find another journal that might suggest different things. So this is something called peer review, all right? So scientists can't just publish anything. There has to be a lot of review. And on top of that, to publish, all right, scientists don't make a profit from publishing their papers. It's exactly the opposite. It costs money to publish. So the paper that I submitted this past summer cost, to get it published with open access, which means that everyone can look at it, it would cost over $2,000, right? So it's not a free thing. Or there's a lot of conditions attached to it, which is what I had to do. I didn't have $2,500 to publish my paper, even though it was accepted, all right? So these are sort of misnomers that a lot of people in the public don't understand, all right? So this is something I just wanted you to think about this. Now, so when these things come out in these scientific journals, all right, the results are only assumed to be valid for the conditions in which the experiment was done. And you always, when you write a paper, you have to suggest what else can be done. What else am I going to do next? Or what can other people do now to sort of validate my work? And scientists love other scientists to repeat their work and add to it, because it makes their whole experience more gratifying and more valid, all right? So if other scientists from around the world are finding the same kind of thing and adding information to it, I mean, that's just icing on the cake, all right? And this is how science works. And if it didn't work this way, then one would have to question the validity of what's being published, all right? So, but this process ensures very high quality information that gets out there. Now, what is the problem with this? Okay, the problem is that the average person doesn't pick up a journal to read. It's not easy to read these things, okay? So, here we have this thing where the author writes it, the editor sends a copy to peer review, it gets peer reviewed, there's accuracy checks, balances, more experiments done, resubmitting, and then we get this wonderful paper, okay? But the thing is, most people can't read it, right? People don't pick up the New England Journal of Medicine. That's probably one you've heard of, but it's not probably something that's sitting on your coffee table at home, all right? So how does the world get their information then? So basically, what happens is that it gets sort of toned down a little bit. We have, and this is sort of a hierarchy of, the reliability of the information that you can find, right? So at the very top, we have that peer-reviewed, refereed kind of information. So that's what you're going to find in these scholarly articles. And then there's going to be other people that are going to then take that information and combine it with other people's and make it, a little sort of reviews on that. All right. Very often those are sort of targeted at academics, okay, or non-academics who are just love to read. All right. But they're easier to read than journal articles. And then we go down, trade magazines, popular magazines. Now the problems with this, and this is what most of us read, all right? So trade magazines, very often, they're going to pick and choose their information because they have an agenda, all right? Or they're doing something for profit. So this is where you have to be careful, right? Popular, all right? So here there's no accountability involved, right? There are some popular magazines that cite the original article, so way up at the top, the refereed and that kind of thing. So one thing that I would give you as an example would be Scientific American. At the end of all of their articles, there's going to be a reference section, and you can go back to those original articles and look at it. So this would be a very reliable type of popular kind of magazine. But other things like People, Time, Newsweek, they all have an agenda, and very often the people that are putting the articles in there are not really scientists, and so things get diluted, and this is what causes a lot of problem. And then right at the bottom, we got the internet, blogs, special interest, things that they have no accountability whatsoever, all right? So, and unfortunately, that's what most of us look at, all right? So what do we do? What do we do to try to cut through this and really educate ourselves, especially on these important issues? And I don't care what those important issues are to you. It doesn't even have to be science, right? Any issue out there that you're reading about, because the internet is everywhere, okay? So here are a few rules, all right? So the first thing is be skeptical, right? Be a skeptic. Don't just take it at face value. Look for evidence. Learn to read the data, okay? Read the signs, okay? And you know what? The numbers are important. Just because something's been done once, that's not good enough. You know that. It doesn't matter what you're thinking about. Even if you're thinking about beetroot juice and making yourself feel better, okay? You gotta look at where you're getting your information. So there are different things here, okay? We've got these different buzzwords that we introduced to you in this course. So we have evidence versus anecdote. What is the difference? Okay, so evidence is a body of facts, all right? Things that you see over and over and over again. An anecdote is like a personal testimony. I'd be like, your aunt so-and-so says, I always have a spoonful of vinegar every morning, and I'm healthy as ever, and this is what you should do. That's an anecdote. That's not good enough, right? Even though you might love your aunt. I love my aunt very much, and she's gone, and then she used to give me all kinds of anecdotes, but you got to think, that's an anecdote. I need evidence. Now, facts versus conclusions. What are those? Facts, separate those from the conclusions. So facts are just pieces of information, right? Sometimes they're numbers, sometimes they're data. They're just pieces of information, but that's not the same as a conclusion. Okay, so if a conclusion comes when you have a lot of facts, lots of numbers, lots of ways of looking at something, and then you can come to some sort of a conclusion. All right, so look at this, and here's a big one, and I'm going to sort of belabor this one a little bit. Correlation versus causation. Correlation, what does that mean? Well, that's a connection between two things. And causation is the act of causing something, all right? Now, this doesn't necessarily mean, if there's a correlation, that doesn't mean that there's a cause, all right? You have to remember something here. When it comes to science, science doesn't care what you want, right? There's no sides, there's no balance in science. Accuracy is the rule here, all right? You can't just say, oh, well, you're biased because you're a scientist. No, that's not the case here. We need to be accurate. There's no, touchy-feelingness here. And I think that's sort of what gets science in trouble these days, because it isn't about that. Science looks at evidence and draws a logical conclusion from that, okay? Now, something that I mentioned in my notes, I didn't put here on my slide, is like if you're looking for information on the web, and even these days, I'm even calling this into question, but it used to be that I could just stand up in front of you and say, at .com, there's always somebody who has an agenda or is making a profit. So look at .gov, .net, .edu, .org. But even now, those four sources, you still have to look for the evidence, all right? Because let's be honest, a lot of .edus, schools get money from different places, and they have an agenda, right? So you have to try and look at what is the agenda that's being followed, and always be skeptical. I know that might sound like a rough way to live, but man, these days you have to do what you need. And this is the example here, all right? Let's think about this. Correlation versus causation, okay? Now, correlation does not mean causation. So here we have on one side, okay, on the right, on the left, we have a graph, and we have the rates of autism, and we can see that those have increased over the years, as well as organic food sales. Now, is that a correlation or a causation? All right, is organic food sales linked to autism? And we did a survey in class, and most people look at me and say, no, can't be. Well, Okay, well, let's look at the other side. We have rates of autism and increased autism with time, and that completely matched as well the increase with vaccination. Okay, now this one, and you know this, there's this big group of people that say vaccination causes autism because of a graph like this, autism and organic food sales. You could probably just swap this around and take organic food sales out and put in vaccination. So what happened because of this? All right. So the idea of vaccination causing autism came from the idea that there was a preservative put into vaccinations called thimerosal based on mercury. Mercury is a toxin, okay, but not in the dose, in the amounts that it was in, anyways. So this caused a big conundrum. And there was a paper published that has been retracted, but there was a paper published that drew a link between thimerosal and autism. And that paper was retracted because the authors admitted to falsifying their data. Now, why they did that, we can talk about more in another time, but they falsified their data. Now, because of that, thimerosal was removed from vaccines. It was removed in 2001, yet the rates of autism have continued to go up, okay? Now, that has precipitated many, many, many thousands of trials and tests to look for some sort of link between autism and vaccination. But the facts are that that first paper was retracted because the scientists admitted to falsifying their data. Fact, the methyl mercury, the thimerosal, was removed many years ago, and autism rates are still going up. Fact, thimerosal does not even accumulate in the brain to cause brain damage. Fact, there's just no links or evidence between autism and vaccination. There's just nothing out there. No one's trying to hide anything, okay? So this is one that has caused so much heartache and damage. If I think about how much money has been spent on putting these two things together that could have been spent on improving treatment of autistic children, I personally find it a little upsetting, right? But this is what can happen when people don't understand the difference between correlation and causation and what makes up the scientific method. So this is something for you to think about. Now, the other thing that I want you to also consider is some of the examples on correlation and causation that are in the book. And one of them is ice cream consumption and shark bites. There's a whole correlation between consumption of ice cream and shark bites. And is this a plausible idea? And of course you think no, and it's because people are more in the water in the summer when people are eating ice cream, right? So, this is something that one needs to think about. Okay, so really look at the data, be skeptical, and think about it, right? And the big thing here with this vaccination and thimerosal and autism, one of the reasons people like to still hold on to this idea is because people don't trust science, right? I mean, we trust organic food, we don't trust vaccines, which is a shame, right? But this is something that we as people need to understand in society and move forward. So what do we do? How can we spot something, even from a .net or a .edu, that is probably false, okay? So there's this thing here, and it's called flares. This is something new that I just came across. I kind of liked it, so I'm going to just talk about it. So now, what does flares stand for? Well, the first thing is falsifiability, okay? So is there any way that the claim can be shown to be false? Logic. Okay, this is where you use your critical thinking skills. Is the claim backed by scientific data? Is there evidence? Replicability. Now, this is a big one. Is there repeated evidence from many sources to back up that claim? All right? And right, S, sufficiency. This is probably the weirdest part of this, but is there sufficient evidence to back up the claim? Think about it. An extraordinary claim needs extraordinary evidence, right? Do you see the evidence? Look at it, right? Is it authority figure or a testimonial, or is it actually data? Okay, and if what you're looking at fails one of these things, it's probably false. Okay, Now, if it passes all of them, well, then it may be true, okay? But if we look at it more critically, maybe it still can be false. But if one of those things isn't there, don't trust it, okay? So just because you really, really want something to be true doesn't make it true, all right? There's all kinds of things that I really, really, really wish were true, but that doesn't make them true, right? Because we're humans, we have passion for what we believe in, right? But try not to fall for that when it really comes down to the wire, all right? Now, this last part, watch a video on this. The big thing, the differences between a hypothesis and a theory. And I already said this in class, right? A theory in science is as close as a scientist is ever going to get to say it's true, okay? This means that there have been many, many hypotheses tested in many, many different ways, so extensively tested in so many different ways, and all of them are pointing to a certain idea, and then we have a theory, a well-accepted answer, well-tested, supported by many experiments by many scientists. And these can take years and years to come, but even theories can be modified. My favorite is that gravity is a theory. I mean, you all believe in gravity, right? You all know how it works. You're going to jump out the window, you're going to hit the ground, right? But even that is a theory because we're still studying gravity. All right, so there's going to be things that are going to be added to our knowledge, even though gravity is a full-on theory. Okay. All right, so that's what I want you to think about for this first chapter. And have a look at the review videos. You might find others. I try to scour the internet for them. You know, you'll find something, but this is what I hope you can get from chapter 1. Okay.